Welcome to this week's episode of the Readout LOUD, a weekly biotech podcast from Stat. I'm a freshly caffeinated Allison DeAngelis.
I'm Adam Forstein, 3 cups of coffee in.
And I'm Damian Garner.
It's Thursday, February 22nd, and here's what we're going to talk about this week.
My brain is buzzing. The drug industry is saying farewell to one of its more polarizing leaders. Stat's Matt Herper and former Alnylam Pharma CEO John Maraganore join us to discuss the legacy of AbbVie CEO Rick Gonzalez.
Then we dive into the fascinating world of fetal genome surgery. Stat's Megan Molteni joins us to explain the work of a scientist named Tippi MacKenzie.
All of that after a word from our sponsor and another cup of coffee.
Hey, Readout Loud listeners, Bob Herman here, Stats Business of Healthcare reporter and the writer behind the newsletter Healthcare Inc. Healthcare Inc. is a weekly newsletter devoted to unpacking the business and secret inner workings of the U.S. healthcare industry. If you're someone who has ever received a medical bill, or craves in-depth policy explainers, or loves a playful meme now and again, I highly recommend you check this newsletter out. Learn more at the link in this episode's description. And now, back to our regularly scheduled programming. Thanks.
After more than a decade as CEO of AbbVie, Richard Gonzalez is leaving the company. Under his tenure, AbbVie's stock price rose almost tenfold. But integral to its success was the autoimmune treatment Humira, a medicine that has dramatically increased its price since its approval, and which escaped competition for many years through some, let's say, baroque applications of the law.
That makes Gonzalez something of a complicated figure in recent pharmaceutical history. Joining us to discuss his legacy are Stat's own Matthew Herper, who wrote about Gonzalez's retirement this week, and former Alnylam CEO John Maraganore, who knows a thing or two about this business. Matt and John, thanks for joining us.
Thanks for having us.
Yes, thank you.
So Matt, let's start with you. In your story this week, you reckoned that AbbVie's commercial success under Gonzalez and all the attendant baggage that made him a lightning rod, you reckoned with that. Where did you land on his legacy?
I think this comes down on some level to what a CEO's job is. Complicated figure is exactly right. I don't think Richard Gonzalez goes down as one of the great pharma CEOs because of that complexity. What I was really trying to square is you talk to investors. I think I've had 3 investors tell me that he is one of the best CEOs ever in the past month. Without me really asking, they brought it up as but AbbVie, that's a company that's really done it right. They've done the transition from I'm confident in the products that are coming along in the future. And then you have this complicated legacy that for people who care about drug pricing, this is a bad example. And to some people, an example of how the system was over-tested and maybe is springing back in a way that's not good for innovation. Kind of an idea that AbbVie's success stole from the rest of the industry and really from the future. Where I landed is that, look, CEOs get fired initially. The first thing that gets you fired as a CEO is you repeatedly don't make your numbers. And if you succeed, you do. And I'm very sensitive to the idea that maybe there should be more. Maybe we want there to be more, but that's the base job. And he did it. And I think that in the memory of people in the industry, he'll probably be more on the plus side than the minus side.
So, John, you posted on X/Twitter a kind of response to Matt's piece, and you said it was way too kind to Gonzalez and AbbVie. What did you mean by that?
Well, I mean, look, I actually think that his legacy will not be as favorable as Matt suspects. I mean, I don't dispute his numbers and the return he generated for shareholders, and I don't know him personally, I must say, but Humira was the straw that broke the camel's back in Washington against our industry. And it ultimately drove the drive to price controls and legislation around that, which will harm innovation and harm patients. And look, one can take a Milton Friedman, capitalistic, or Ramaswamian view that the CEO's sole job is to make money for shareholders. But I believe that the biopharma CEO's job is to make medicines for patients and from there, reward shareholders, the George Merck famous saying. So that's where I disagree with Matt's take on his legacy at the end of the day.
So, Matt, let me ask you. So, John is sort of drawing a direct line between Humira, the pricing of Humira, the patent estate around it, and the Inflation Reduction Act and all the drug pricing kind of stuff that's going on now. I mean, do you agree with John? I mean, what's your take on that?
I actually think John and I agree more than we disagree in some ways, but I think the fundamental area of disagreement here is that I would like all CEOs to be like George Merck or Ken Frazier or John. I don't think that is, after years of covering and watching CEOs, that is the first part of the job. That's the part of the job that makes figures truly legendary in an industry, when they're able to transcend just making the numbers. There's a contrast here and a conflict here that's really, I mean, it's not just what's difficult about pharma, it's what's difficult about the world, right? And I would like it to be true that pharma CEOs are judged on their impact on the larger world. And I think they are to some degree. But I think that from the perspective of the people who hire CEOs, who are in essence shareholders and boards, that's not the case. That they're quite happy to take someone who delivers the returns, and quite a lot of CEOs don't.
Well, I think, Matt, I have to agree that delivering returns is an important and critical part of a CEO's job, and we took that very seriously at Alnylam as well. But it really was just the approach for how you would do it, and I do believe that a biopharma CEO's job is to make medicines for patients, and if you do that, that's the beauty of it. If you do that well, then you will reward shareholders, because it naturally follows. Those profits naturally follow, as we know from the beauty of innovation and how it makes an impact at the end of the day. If you take the shorter term view and say, I'm going to milk this cow till I get every piece of milk, every drop of milk out of it, and I could give a crap about the rest of the sector and the long-term implications, that is not a great way to ultimately create an ecosystem that is sustainable at the end. And that is where I take issue with Rick's legacy in this regard.
So, John, do you think is it possible to do both, to accomplish both,
Yes.
to both generate a return for shareholders and kind of think about the broader world about in terms of drug pricing and access, and sort of not do what Gonzalez and AbbVie did around Humira?
Yes, I totally think it is. I mean,
but John, I have to play Ian Reed actually a bit here, because I remember there was a long time ago I was party to a conversation between Ian and Len Schleifer where they kind of had this argument. And the other argument there is that it's very easy for someone who's run an early stage biotech company to say that. But it's very uncommon for someone who is running a company that has a $10 billion, $20 billion drug driving its profits to not look for every opportunity to prolong. It's a very difficult thing to ask of a board, and your fiduciary duty does end up coming into direct conflict with making the ecosystem you want in the future. There's a choice that has to be made, and I don't know that there are any CEOs who are celebrated for making the choice that they were just going to let it expire, that they were just going to let it go. I am sympathetic to the idea that this went too far and broke the industry's back, but I think the question is, was it his job to save everybody else?
Matt, it's interesting. I remember that conversation between Ian and Len. And it was around price increases that were going on from Pfizer, and Len was commenting on it. The issue on all this is really the productivity of the industry at the end of the day. And you had Len. It's not about biotech versus pharma. It's between Len, who's got an amazing platform that generates sustainable innovation, versus the challenges that existed at Pfizer at the time, where there wasn't a product engine that delivered a sustainability around their growth. It was really around the fundamental productivity of their R&D engines at the end of the day. And that's why one company, namely Regeneron, was able to grow organically, sustainably, without annual price increases. And the other company, Pfizer in this case, had to rely on other tricks of the trade. And I think that's the issue, not necessarily having to deal with a $20 billion baseline per se, but really the real productivity of R&D.
But John, is there a way to set the world up so that shareholders actually want that? Because I guess in 20 years of watching this, my impression is that the investor base doesn't always care, right? I mean, they loved Mike Pearson at Valiant, who was probably the ultimate example of let's not do any R&D. We'll buy stuff. We'll raise the prices. We'll make tons of money, right?
Yeah.
How do you say somebody who seems to have built a sustainable company, made smart acquisitions, very hard to do when facing a large patent expiration? How do you say that they didn't do the job of the CEO just because you feel they didn't do this larger societal job of being a leader in society as well as being a leader of their company?
Well, again, it's about the sustainability. It ultimately comes down to near-term reward versus mid to longer-term reward. If you poison an industry in a way that ultimately erodes the industry's ability to be successful, longer term, you have not done the right job for shareholders. I don't think we disagree with that, right? So it's really a near term versus longer term issue. And I think a CEO should balance both sides of that equation at the end of the day. I mean, we all think about strategy and growth of our companies, and we think about it in the context of what are the near term things I've got to do, and then how do I look at my mid to longer term prospects? Well, a CEO has to think about that more broadly, not just within the context of their specific company, but also the ecosystem that they live within.
Let me pose this question to both of you. Gonzalez, he's retiring. He's going to be gone. So what lesson do you think that the industry is going to learn from his departure? I mean, which direction do you think it will go? Is it going to go in the Rick Gonzalez direction, or is it going to go in the John Maraganore direction?
Well, I hope, look, I because I've retired, I hope it doesn't go in my direction, and Rick's retired too. But I do hope it finds a balance between these two extremes. And I think that there is an answer in the middle. And look, we've got some great CEOs in the industry, including in big pharma. Look at Dave Ricks and Albert Bourla, others, that are really, some of them are struggling. But they are great CEOs. They think about the near-term issues and objectives, and they think about the longer-term objectives as well. And I'd love to see a lot more of that in the future leadership of the industry.
I hope that's what we see. But I really think that the pressure on CEOs often pushes them much more in the direction of holding on to profits than thinking about the larger needs of, or the position of the industry in society.
Yeah, that's true.
With the kind of legacy, this connection of Rick and Humira's influence on the IRA being passed, that linkage that you two are both talking about, in kind of a post-IRA world, in a post-IRA biopharma industry, is there even room for kind of the playbook that Rick Gonzalez's career used?
Allison, I think there are different versions of the playbook that companies will adapt to, even with the IRA in play, right? And hopefully that, who knows what business innovation will emerge from all that in the future. But I still think the CEOs need to balance, in our industry, need to balance the near-term objectives with that mid to longer-term ecosystem impact of what we do.
Matt, John, thank you both. I love seeing a Twitter conversation get brought over into the audio sphere and kind of expanded. And yes, I'm always going to call it Twitter. I'm never going to call it X. Thank you both for joining us today.
Thank you.
Thank you, guys.
It's a situation that expectant parents dread. A routine checkup finds that the child you hope to bring into the world has a life-threatening genetic condition that can't be solved with surgery. There are often few options but to see if the child survives to birth and is strong enough for treatment, if there's even one available.
For years, the conventional wisdom has been that in utero therapeutics would likely never be a reality, both because of practical barriers and then ethical ones. There are those who argue that even if it were possible, it shouldn't be attempted. Our colleague Megan Molteni profiled one woman who hopes to overturn those notions. And Megan joins us now to discuss this scientist's work. Welcome back to the podcast.
Happy to be here.
So Megan, let's set the stage for listeners. Your article dives into what's called fetal genome surgery. What is that, and how does it compare to other uses of CRISPR?
Sure. And to be clear, fetal genome surgery is sort of a term that I came up with for the purposes of explaining how
It's
this works.
coined now,
Because
so
It's a good one.
we're running with it.
We're setting the trend.
in utero genome editing is like a little bit of a mouthful. But essentially what we're talking about is sort of like the next evolution within the field of fetal surgery. So this started in the 80s with the tools of the trade. You had scalpels, you had sutures. That sort of has moved to more laparoscopic technologies, much less invasive things. And then you sort of have what are, now we would call, sort of molecular therapies. So you might have an enzyme that you want to give to that fetus because that fetus is not making it itself. And because of sort of the unique uterine environment, the way the mother and developing fetus are connected at that time, it's actually a really simple procedure where you simply inject directly into the umbilical vein, and that is able to reach the fetal blood system, sort of go throughout its developing body. And so you can deliver things, what have been done so far in clinical trials are delivering enzymes. They've also done stem cell transplants in this way, sort of same procedure. That has been less successful, but it's sort of building on the fact that for many decades, fetal surgeons have known that you can do fetal blood transfusions this way. So this is just sort of taking an existing sort of evolution in this medical field and adding CRISPR, since it is obviously the sort of most exciting tool that we have going on right now. And so infuse that into the umbilical vein and have that go into the fetal cells and do the work it's going to do on the DNA there. And so sort of fetal genome surgery, I think, captures the idea that you're sort of within the field. The field of medicine that you're in is very much rooted in fetal surgery, fetal medicine. These are doctors these are their primary patients, and you're just kind of adding CRISPR.
Now, the idea of mixing fetuses and CRISPR, and CRISPR in babies, is not new. Obviously, in 2018, there was a rather big event in China that kind of set the scientific community ablaze. How does the community feel now about the prospect of in utero genetic editing compared to the embryonic editing that was conducted in 2018?
Yeah, well, just to go back to, you said it's not new for CRISPR and fetuses. And I just want to, really clarify on this because this is a really important point. When we're talking about the Chinese experiment in 2018, those children that resulted from that experiment were edited as embryos. So IVF embryos in a dish when they're sort of a single cell or a couple, actually in that case, it was 1 to 2 couple of cells old. And then they developed, sort of all the way through to birth. And as far as we know, are, children at this point. But what Tippi MacKenzie, who is the surgeon that I profiled in this, what she is proposing is a very different procedure. So this would be editing a fetus in the 2nd or 3rd trimester at the point at which you're able to go in with genetic testing, with other kinds of molecular testing, and actually get a diagnosis so that you know what's wrong with that patient. And the distinction is important because from an ethical standpoint, the way bioethicists think about that is that's essentially just conventional medicine. You have a patient, they have something wrong with them, and you have a treatment that you can give them. You're just administering that treatment prior to birth at a time when the treatment has a higher likelihood actually of being more effective because of the way, CRISPR and other therapies can really like spread throughout all the cells of a developing fetus. There are cells that you can't reach in a child after they've been born that you can in the uterus. So the hematopoietic stem cells, these are the cells that are treated in sickle cell disease, that are treated with Casgevy. Right now, they have to go through a process where they release those cells from the bone marrow. They pull them out, they collect them, they treat them in the lab. They have to go through chemotherapy to then, get their bone marrow emptied out to then receive those cells. And it's a really, it's just a really rough process for the patients. And in this case, those hematopoietic stem cells are actually living in the liver in a developing fetus. And so you can just reach them much more easily. And the same is true for neurons and other brain cells that aren't yet sort of locked behind the blood-brain barrier. So neurodegenerative diseases are particularly, potentially amenable to this approach. And that is different conceptually than having an embryo that you don't know anything about that embryo. There's no, you might know its genome, but you don't know anything about how it's developing. You don't know, if it's developing an abnormal heart condition because of the genetic sort of disposition behind it. You don't really have, what they would call a phenotype, something to really diagnose that you're changing. I mean, in the case of the Chinese experiment, as far as we know, there was nothing wrong with those embryos. They were edited to make defective a receptor that is what the HIV virus uses to get into cells. And so the idea was to sort of make them resistant to HIV. And that's a really different proposition, because in the one case, you have a patient that has a disease, and if you don't do anything, there will be suffering and probable death. And in the first case, in the case of embryos, the intervention is kind of the point.
So getting to MacKenzie's career, I mean, one thing that was fascinating to me about both your story and your description of this approach just now is in itself the word surgery. I think when we talk about CRISPR, both in the culture and even at the FDA, we talk about it like a therapeutic, like a drug. It's something that's administered. Whereas I totally understand where the application here, at least in the words of MacKenzie and others, is much more like that of surgery, where it's a procedure. It is something that's done. And as you illustrate in your story, her work now has roots in the old-fashioned kind of surgery. Old-fashioned is maybe a little bit pejorative, but the surgery, the processes we think of when we hear the word surgery involving scalpels, et cetera, as you mentioned. So could you I know the story is very long and people should read it, and I'm not asking you to recapitulate it in like 30 seconds, but when did you first learn about her, and how does the lineage of fetal surgery as it has been practiced for some decades link to this very modern take on it that uses CRISPR to do surgery at the molecular level?
Yeah, so I first met MacKenzie in London last year at the sort of third International Genome Editing Summit. So this was the summit that took place sort of following the one in 2018, where the Chinese experiment sort of details were revealed, and where sort of that stain on the field was sort of very much still present. It was very much part of the programming while we were there. And what really struck me and why I wanted to spend some time with MacKenzie is that in the genome editing field, you sort of like people who are genome editors, like they build tools. Like often there are folks who, are also physicians, but they're really sort of building these molecular tools that can cut DNA, that can add DNA, that can do, they're more like, the producers of like bespoke scalpels. And when I went back and looked at sort of the history of fetal molecular therapies, I was surprised to learn that almost immediately after some of the first human tests of gene therapy, that those people who had developed the tools of gene therapy, so we're talking about, transgenes and viral vectors, they were like, let's go in utero right now. They were like going to the NIH, RAC, they were like going to the FDA. And it was really being driven sort of by the tool developers, by the people who were like, made these gene therapy vectors. They're like, this is awesome. It could be even more awesome to treat fetal patients. Let's do this. And that, the history of gene therapy, it was obviously pretty, disastrous early on. And it really, I think, has a lasting legacy in the genome editing community, where the folks who make the tools are pretty cautious about how those tools get applied and who the populations are, who they want to start doing these first tests with. There's a way in which MacKenzie coming to it from a totally other discipline, coming to it from being a surgeon who sees fetal patients on the regular, she just has a really different sort of way of thinking about, what sort of acceptable risks are, what transparency looks like, what patient autonomy looks like, because she, her whole career, she has essentially treated sort of two patients at the same time, right? She's essentially asking, the person carrying that fetus to accept all the risk of that surgery, of that procedure, on behalf of the child developing inside them. And so she's really steeped in what would that mean. I think like the genome editing community is really excited to have someone like that who's really, really responsible, really thoughtful, as sort of being the person leading this charge, because of sort of the history of what happened in China and some of the early excitement with gene therapy that was really unwarranted given where the science was at the time.
And Megan, help us better understand what those potential risks are, when you introduce a CRISPR therapy into a pregnant woman and a fetus.
So like I said, you have two patients, and one of them, the person who's carrying the fetus is taking on all the risks and doesn't stand to have any of the benefits. The fetus is going to stand to have all the potential therapeutic benefit. So there's primarily a concern with risk to the mother, and that would essentially encompass, can some of these CRISPR molecules, depending on how it's delivered, cross over into the maternal bloodstream and either cause an immune reaction or, do some editing of her cells, which who knows sort of what the impact of that would be. But those are, I think those sort of pale in comparison to the risks of, what the fetus would be assuming. So, sort of like a double-edged sword of this developmental stage where more and different types of cells are accessible. And so you can treat things that are much harder to treat after birth. But that also means that these, genome editing components can really be distributed widely across different tissues. And the biggest concern right now is that some of the tissues that might be inadvertently impacted and edited would be germline tissues. So that would be cells that become sperm and become egg. And this is a concern, not so much as we don't know what the health risk would be to the fetus, but there's sort of a very broad global consensus that germline editing is really sort of a no-go at this point, because we just don't know what the impact is going to be of passing down these changes to future generations. And here in the US and in like 70 other countries, that would be enough for a regulator to say, absolutely not, you can't try this in humans if there's evidence of that. The thing that you're trying to do is find a window in development where tissues are accessible enough that you can edit them, but tissues like germline are not. And there may be ways to sort of tweak that on a tool molecular level to say, only activate these genome editing components in certain types of tissues if that have this type of, cell marker, but that's still off in the future.
Well, on that final point, as you mentioned, MacKenzie is very clear that there's a lot of work that needs to be done before she or really anyone would be prepared to make the case to the FDA to allow for a first clinical trial of one of these attempts at fetal genomic surgery. But can you give us, I mean, I know that all of these answers, kind of demand a lot of detail. And so I'm asking you to sort of commit the sin of yada, yada, yada-ing some stuff. But in your sense, what kind of work is necessary to get to that point where testing this in a human trial is potentially viable, and how long might it take to get there?
I will say that there are a few other labs that are also pushing this forward, including with support of the NIH, and they are, they're doing experiments in mice and in non-human primates. And the way they are thinking about it, this is Bill Parento at CHOP and Kieran Musunuru at University of Pennsylvania. And the way they're thinking about it is that they don't believe the FDA is going to greenlight an in utero CRISPR therapy that has not yet already been approved and has a couple of years of follow-up data in postnatal populations, so in kids and infants and adults. I think what the FDA really needs to see, first and foremost, is no evidence of germline transmission. And so that would mean that you're using you're doing tests with the same clinical grade treatment that, has been approved postnatally, and you are testing that in large animal models. And in this case, the fetal model they use is sheep. And then you're looking at, where did it go? Did it edit in germline cells? And so, those experiments are expensive, and they take a couple years to get set up. There's also the issue of finding sort of the disease, the sort of first use case disease where it's serious enough that this is, that it would warrant this kind of approach. You sort of need to be talking about diseases that are otherwise fatal in utero. And, that's why no one's trying, Casgevy in utero right now, right? Because the risk benefit for something like that just doesn't net out at this stage. So there's some work to be done to figure out what exactly, is that right disease use case. But I think really the biggest issue for regulators is going to be the germline issue. So I think that's where her efforts are focused at this time.
Well, Megan, this is a fascinating topic, and I encourage all of our listeners to go to statnews.com and read Megan's profile of Tippi MacKenzie and her work in fetal genome surgery. Megan, thanks so much for joining us.
Yeah, thanks for talking about this. It's just really wild. It's really cool.
That does it for another episode of the Read Out Loud.
Thank you to Teresa Gaffney for producing this week's episode.
Our senior producers are Hyacinth Empanato and Alyssa Ambrose. Our executive producer is Rick Burke, and our theme music is by Brian Joel.
We'd love to hear from you. Tell us what you like about this week's episode, what you didn't like, and your thoughts on Rick Gonzalez's legacy. You can do all that by sending us an email at readoutloud@statnews.com.
And if you like what we do, leave a review or rating on Apple Podcasts or whichever platform you use to get your podcasts.
See you next week. Sorry. Cut this out of the... Keep everything else. Cut this out.
Let's leave that in. Let's leave that in.
Like
leave the whole thing in.
Like this is some weird biohacking project for you. Like you begin really high on whatever, then it's just suddenly choking to death seconds later.